Welcome to the new research media briefing of the Brain and Behavior Research Foundation. I'm Dr. Jeff Borenstein, President and CEO of the Foundation. The Brain and Behavior Research Foundation, formerly known as NARSAD, raises funds to support cutting-edge research projects to understand, treat, and ultimately prevent and cure mental illness. Since 1987, the Foundation has awarded nearly $300 million in research grants to more than 3,300 scientists around the world. Research projects are selected by the Foundation's Scientific Council, comprised of 138 leading experts. The Foundation invests 100% of donor contributions for research directly into research grants. For more information, please visit bbrfoundation.org. This is a very exciting time for brain science. As many of you are aware, the President is moving forward with his brain initiative. There have been extremely important findings that have been occurring over the last period of time. And I'm delighted today to be your host of this event. As Editor-in-Chief of Psychiatric News and Chair of the Communications Council of the American Psychiatric Association, I've moderated a similar type of event at the APA's annual meeting, and we've borrowed the format for today. At today's conference, honorees will present about the body of their work as well as recent and preliminary findings. Each scientist is asked to provide any disclosure information as well. I will briefly introduce each person, but refer the reporters to our news release link on each slide for further details. Questions will be addressed at the end. To submit a question, use the Questions tab of the control panel and specify the researcher whom you wish the question to be addressed to. We will answer as many questions as possible. If we don't get to your question, please email Sally Corbett, SCORBETT, at bbrfoundation.org for further assistance. Today we're featuring 6 NARSAD Young Investigator grantees who will be honored Friday at the Brain and Behavior Research Foundation's 19th Annual Klerman Prize and 16th Annual Freedman Prize ceremonies. The Klerman Prize for Exceptional Clinical Research was established by Dr. Myrna Weissman in memory of her late husband, Dr. Gerald L. Klerman, a distinguished psychiatric researcher at the NIMH and member of the faculties of Yale, Harvard, and Cornell universities. Dr. Klerman served from 1977 to 1980 as Chief Administrator of the Alcohol, Drug Abuse, and Mental Health Administration. The Freedman Prize for Exceptional Basic Research honors the late Dr. Daniel X. Freedman, a pioneer in biological psychiatry and psychopharmacology and a founding member of our scientific council. We will begin with the 2013 Klerman Prize winner, Dr. James McPartland of the Yale Child Study Center. Dr. McPartland is an assistant professor of child psychiatry and psychology and director of the Developmental Disabilities Clinic at Yale University. He is being honored for his discovery of a novel electrophysiological marker of eye contact that predicts social ability in children. Dr. McPartland.
Thank you so much. It's a pleasure to have the chance to speak with you today. I want to talk briefly about 2 things, really the way that my lab approaches understanding autism, and then some of these preliminary findings that we're very excited about. So I am a clinical psychologist by training, and that is really influential in the way that I approach autism. I've spent many, many hours with children on the spectrum and with their families, and it's very important to me to guide my science and also to try to understand what is important to families to address. I think that our clinical insights are key, but they're not sufficient. We need ways to understand mechanisms. And so we try to pair these clinical insights with neuroscience. And particularly, we're very interested in developing methods to study brain activity related to the really nuanced social difficulties that we see in children on the spectrum. And the hope is by combining these clinical insights with these neuroscience methods, we can make a difference, that we can improve the lives of children with autism and their families. I'd like to talk about a really particular facet of our research, trying to look at eye contact. We think of eye contact as really the basis of social interaction from the very, very first days of life. And it's also a key problem in autism that's evident very, very early in development for children on the spectrum. We think for these reasons that it's really central to improving diagnostic methods and treatment methods for this social disability. Although these clinical insights lead us to eye contact, it's a very hard thing to study as a neuroscientist because eye contact exists only in the context of interaction. It's something that happens between two people, and most laboratory studies aren't interactive. What we've tried to do is combine two methods that have both been very helpful in understanding autism: eye tracking to measure where a person looks, what they look at, and EEG to measure brain activity with really precise temporal specificity. And what we've done is integrate these two methods so that we can have experiments that measure brain activity but respond to what a person looks at. Specifically, we have faces on screen that are responsive to the person watching them. This lets us measure brain activity in an interactive context for the first time in autism research. As you can see on the right, you see a blue dot indicating where a person looks. And in this experiment, faces look back at a person when he or she looks to them, and they smile when they make eye contact. And so our hope by building this kind of experiment is that we could see something different, something interactive. And we think it's paying off. This chart shows a waveform, the EEG activity, the brain activity of a group of typical children. And what I want you to focus on is this peak. This peak, it occurs very quickly, about a quarter of a second after the eyes lock, but this is the brain of typical children registering that moment of connection to the eyes. And in the children with autism that we've seen so far, and these are preliminary data, we don't see this response. We're very excited about the potential of this marker to improve our standards of care. Because eye contact is evident so early in life and is a problem for autism so early in life, we hope that these tools can be applied for early detection. We want to know, by looking at these markers of brain activity, who is likely to respond to treatments and who isn't, so we don't waste time treating people with interventions that aren't indicated. And we want to understand why some people do so well and some people struggle, even with the best interventions. And we hope that this marker can help us do that. I want to close by acknowledging the support of the Brain and Behavior Research Foundation. As a clinician, having the opportunity to have protected time to conduct research has been so critical for my career. In keeping with the idea of these Young Investigator Awards, it's also been so helpful in gathering data to be able to garner additional support to do more research. And so as you can see from the slide, we've had lots of opportunities now to build a program of research, and we're very excited about it. So thank you so much for the chance to speak, and I'll look forward to answering questions.
Great. Thank you, Dr. McPartlin. This is extremely exciting research that can have a tremendous impact in people's lives. So I'm sure we'll have some questions about it, and thank you for the work that you're doing. Next, I'm pleased to present the 2013 Freedman Prize winner, Dr. Garrett D. Stuber. Dr. Stuber is an assistant professor of psychiatry and cell biology and physiology at the University of North Carolina School of Medicine. He is being honored for his research exploring the role of dopamine and non-dopamine neurons in the midbrain. Dr. Stuber.
Great. Thank you very much for the opportunity to discuss some of our recent work. My lab, one of the major focuses of my lab is studying the neural circuitry that is responsible for the manifestation of very sort of complex motivated behavioral states. And so in the lab we have a couple of goals that we're working towards. The first is to really identify and to characterize the precise neural circuits within the brain that control very specific aspects of motivated behavior. And when I'm talking about characterizing these neural circuits, we're really interested in understanding the genetic definition of these cells, what makes them different from neighboring neurons, what cells are they connected to, how are they functioning differently. We think that if we can understand how neural circuit function is altered, we can use animal models to look at this to see how in animal models of mental illness such as anxiety or depression models, we can understand how these circuits may be altered in humans as well. And really we think that by elucidating these circuit targets will provide really critical information for the development of future pharmacotherapies, because we'll understand exactly what precise circuits or neurons or connections to try to study and further and to try to develop therapies to target. So how do we go about manipulating neural circuits in experimental contexts? This is a really complicated problem because the brain is such heterogeneous tissue. It's the connectivity and complexity is enormous, so it's not as simple as going into a specific brain region and lesioning it or electrically stimulating it. Each brain region is composed of a number of different genetically defined cells that are all interconnected in very complicated patterns. So what we'd like to do is to be able to use technologies that allow us to essentially disentangle the brain and to study it in a more highly linearized specific fashion, where we can manipulate one particular neural circuit node and see an output that occurs either on the physiological level or on the behavioral level. And so how we go about doing this, we use a number of contemporary tools within systems neuroscience to do this, but one that we've used extensively over the past couple of years is using optogenetics, where we can introduce engineered proteins that are light sensitive that can be used to control the activity states of neurons. So typically we can introduce these proteins either using a viral construct or in transgenic animals, and selectively express these proteins, these opsin proteins, in specific populations of neurons. And when we shine light into the brain, light of a particular wavelength will activate these proteins, cause a change in their conformation, which will then depolarize or hyperpolarize to activate or suppress the activity of the neurons that are expressing these. And so the real benefit of this is that we can use techniques from molecular biology and genetic engineering to selectively express these proteins in a brain region in a particular subpopulation of neurons, apply light, and selectively activate those cells without directly affecting the activity states of neighboring cells that would not be targeted. And this allows for real precise specificity for neural circuit manipulations. So over the past couple of years, one of our lines of research in the lab has been focusing on a couple of interconnected brain regions that we think are very critical for the manifestation of a number of different neuropsychiatric illnesses. One of these areas is called the lateral habenula. It's known that this is an area of the brain that becomes hyperactive or overactive in neuropsychiatric disease such as depression. And we're also looking at the connectivity from the lateral habenula to other brain regions, in particular the ventral midbrain, which when I'm talking about the ventral midbrain, I'm talking about both the ventral tegmental area and this sort of posterior extension of this called the RMTG. The ventral tegmental area is a brain region that contains dopaminergic neurons, and of course probably everybody's familiar that dopamine is a neurotransmitter that when released promotes reward-related behaviors, positive reinforcement, motivation, etc. And so what we're interested in looking at is this interconnectivity from the lateral habenula to the ventral tegmental area in neighboring regions. So in the first study that we published last year, what we did was to introduce chanorodopsin, which is an opsin that can be used for activating particular neurons, into these lateral habenula neurons that project to the RMTG VTA. And this is an excitatory projection. We then performed a number of different behavioral experiments that we could selectively activate this pathway from these neurons. And then we used simple behavioral tests coupled to stimulation of this pathway to determine what are the behavioral consequences of selectively activating this pathway. And this is just an example video of what we saw. Hopefully you'll be able to see it. Essentially, this is an animal that's not expressing an opsin, and we look at essentially where it's spending its time in a chamber. When it's on one side of the chamber, it receives stimulation. When it's on the other side, it doesn't. Here's the active animal that's expressing chanorodopsin, and what we found was that you can see when the laser turns on to activate this projection from the habenula to the midbrain that the animal clearly avoids activation of that chamber. And so this is just a summary of that data showing that they significantly avoided the chamber that was paired with stimulation of this projection. More recently, in an unpublished study, we've also looked at this projection back from the VTA, which also innervates the habenula. So this is, I just talked about this projection going from the lateral habenula to the VTA RMTG. We've more recently looked at this reciprocal projection, and what we see is that this produces essentially the opposite behavioral effect in that the projection from the VTA actually shuts down the habenula, which can then produce reward-related behaviors. So it seems that the dopamine projection is actually actively suppressing the activity of habenula neurons to promote reward. And so just to conclude, what I just talked about was that neural activity from the lateral habenula to the ventral tegmental area promotes aversion in mice, and we think this is done by the indirect suppression of dopamine neuron output. There's a reciprocal connection that exists between the VTA that projects back to the lateral habenula, and that activity in this pathway promotes reward-related behaviors by inhibiting the activity of those lateral habenula neurons. And we think that this functional connectivity that I've just described to you provide novel neural circuit targets for potential therapeutic intervention for the treatment of neuropsychiatric illnesses. Finally, I'd just like to acknowledge the people in my lab who did this work. Most of this work I just described was done by a graduate student, Alice Damatakis, in my lab, in collaboration with a couple other people in the lab. And finally, I would like to thank our funding agencies for the support, in particular the Brain and Behavior Research Foundation, who my NARSAD Young Investigator Award was really critical for getting these early studies up and running in the lab. I'd also like to acknowledge all of our other funding sources that have contributed to this. Thank you for your attention. I'm happy to take any questions.
Thank you. Thank you very, very much, Dr. Stuber. Very important work. And just as an aside, to put a little bit of context into the importance of these young investigator grants, the technique optogenetics that Dr. Stuber is using in his lab is being used around the world right now in many, many labs, thousands of labs. And this is a technique that was developed by a person who was then a young investigator grantee, Dr. Carl Differoff, who is currently at Stanford, and who subsequently received an Independent Investigator Award and a Distinguished Investigator Award, and is now a member of the Foundation Scientific Council. So it just is an example of how young investigators are able to make use of the support from the Foundation and have tremendous impact. Next, we hear from Klerman Prize Honorable Mention recipient, Dr. Andrea Dunazzi. Dr. Dunazzi is an associate professor at the Institute of Psychiatry, King's College, London. He studies stress and stress-related psychopathology in young people, focusing on the biological mechanisms through which early experiences influence child development and exert lifelong effects on health. Dr. Dunazzi.
Yes, hi. Thank you very much for the opportunity to discuss my research today. My research has to do with stress in children. Is based on the evidence that when we are exposed to threat, our brain triggers bodily defenses, for example, producing blood molecules that protect us from infections, the so-called inflammation protein. In the past, I have found that childhood threats, like maltreatment, have enduring effects that persist well beyond exposure to threat. Maltreated children grow up to have high levels of inflammation proteins in their blood when they are adults. And because of these enduring effects, as you can see portrayed here, depressed adults with a history of childhood maltreatment have elevated inflammation levels when compared to controls, while depressed individuals without a history of maltreatment do not. These are data based on prospectively collected information on maltreatment from the Dunedin Multidisciplinary Health and Development Study. In addition to being enduring, these effects seem to emerge early in life. As you can see in this set of slides, already at age 12, in members of the E-RISK environmental risk longitudinal twin study based here in the UK, already at age 12, depressed children with a history of maltreatment have elevated inflammation levels compared to controls, whereas in this pilot study, depressed-only children, so children with depression but no history of maltreatment, do not show this elevation. I believe these are not only interesting biological findings, but may have important clinical implications. This slide, for example, shows that inflammation levels are elevated here in individuals with recurrent episode of depression, but not in individuals with single episode of depression. Furthermore, and consistent with the epidemiological findings, several clinical trials now are showing that inflammation levels are elevated at baseline in individuals here that are resistant to treatment, but are similar to controls in those who are responders to treatment. Therefore, I reason that on the one hand, maltreated individuals tend to have elevated inflammation levels, and on the other hand, elevated inflammation levels are associated with recurrence and treatment resistance in depression. I was then curious to see if maltreatment predicted recurrence and treatment resistance in depression. And for this reason, I carried out a meta-analysis. That is, I pulled together all the studies looking at these very questions and weighted the results based on their quality. You see here these blue boxes are graphical representation of each single study in the meta-analysis. The line in blue being errors related to those studies. Red diamonds here are the cumulative effects of these different meta-analyses. And what you can see is that studies that overall effect sizes that do not overlap with this risk equal to 1 are significant. And the meta-analysis therefore shows that maltreated individuals are at elevated risk compared to non-maltreated individuals to experience both recurrence and persistence in depression. And these are the forms of depression that account for the most of the personal and economical burden of depression. In addition, I found that depressed individuals with a history of maltreatment are also at elevated risk of treatment response. This is the overall results looking at different types of treatment for depression. And what you can see is that maltreated individuals have particularly poor response when pharmacotherapy or combined psychotherapy and pharmacotherapy are used for the treatment. That is, the most powerful treatment for depression seems to fail to help this vulnerable group of people who go on and develop recurrent and persistent episodes of depression. Therefore, we need to find new forms of treatment. And building on my research and on research of other colleagues interested in the interplay between brain and body, a growing body of literature suggests that targeting inflammation might just help to fill this treatment gap. For example, in this study, researchers have looked at a group of treatment-resistant cases of depression. And what they have done was stratifying, so dividing the group in those with high levels of inflammation on the right-hand side and low levels of inflammation on the left-hand side. And what you see is that adding an anti-inflammatory medication, this infliximab, leads to significant reduction in responders, increase in responders in those with elevated inflammation levels. So overall, this research suggests that if we understand more of the biological correlates of child maltreatment, we may be able to find new cures for depression and other psychiatric disorders. And I wanted to end by thanking my collaborators from the Dunedin Multidisciplinary Health and Development Study in Dunedin, New Zealand, and the Environmental Risk Longitudinal Twin Study here in UK, and thank, of course, the Brain Behavior Research Foundation for their generosity in funding the early critical stages of my research. And I thank you for your attention. I will be very happy to take any questions. Thanks.
Right. Thank you very much. Let's move on with the next presentation. That will be Klerman Prize Honorable Mention recipient, Dr. Daniel Muller. Dr. Muller is head of the Pharmacogenetics Research Clinic at the Center for Addiction and Mental Health and an associate professor of psychiatry at the University of Toronto. The goal of his research is to improve psychopharmacologic treatment through personalized medicine. Dr. Muller.
Yes, thank you very much for this nice introduction. And here is my presentation. I hope you can see the first slide. This is basically about pharmacogenetics. And when I was a resident, I was stunned by the variability, first of all, of the medication that were used to treat patients in psychiatry. And I was already doing some genetic research in bipolar disorder and schizophrenia, but I became more so interested then in trying to identify genetic factors associated with treatment response and drug metabolism side effects. So this is one of my topics, which is the genetics of antipsychotic-induced weight gain, the project that was funded by the NARSAD Young Investigator Award. So let me see. Why can I move on here? Oh, let me see. Okay, good. So here is a just a brief graph to demonstrate the amount of weight gain that can be induced by some antipsychotics on average. It's about 5 kilograms, 4 to 5 kilograms after 6 to 8 weeks. And this weight gain, again, is just an average measure. We see huge variability. Some patients gain 10 or 20 kilograms within 6 to 8 weeks and may continue to gain weight beyond 6 months or 12 months and can really often get severely obese, leading to all the secondary complications such as diabetes, cardiovascular disease, and premature death. And two medications here that are particularly important to mention are olanzapine and clozapine, because these medications are basically working very well in schizophrenia often, and there's often no alternative to use these two drugs, unfortunately. And there is good evidence by twin studies and first relative studies to believe that there is a genetic component in antipsychotic-induced weight gain, as evidenced by the study above. And it also has become quite prominent or quite clear that this weight gain is often a result of binge eating behavior that is induced by these medications. So a first thought is, if you look into the genes, if you want to find the genes, is what are these drugs doing that may interfere with appetite regulation. And one of the candidates is the 5-HT2C gene, which is shown here. And we've done some of the studies listed here in this graph and done this meta-analysis. And even though the sample size are relatively small, particularly for genetic studies or what we currently see, we were still seeing a strong overall effect for one polymorphism of that gene, and with an odds ratio of 2.4, which is quite good also considering genetic findings in complex disorders in general, where odds ratios are often far lower than that. And if we refine this also for early weight gain, we see even larger effects. So the 5-HT2C seems to be implicated particularly in the early stages of antipsychotic-induced weight gain, which could be used in a predictive test in order to identify who is at high risk for weight gain, and if this test, together with other genes and other factors, may become available, that would be very helpful in order to basically prevent weight gain in some patients or treat patients who have low risk for weight gain with medication that they may benefit from. So the last years, and that was particularly the one, the studies sponsored by NARCET, we were then interested in moving on from the receptors that are targeted by antipsychotics and look into the hypothalamic pathways, which are key players in appetite and satiety regulation. And these are just a few examples of key players such as the neuropeptide Y gene, the BDNF gene, the melanocortin-4 receptor, and the leptin genes. And interestingly, in all of these genes, we found some signals indicating that genetic variation in these genes are playing a role in antipsychotic-induced weight gain. This is demonstrated here. This is the hypothalamus, and if you look at the middle, you'll see the MC4R gene, which was supposed to be circled. Unfortunately, here it didn't work, I think. But anyway, if you keep the MC4R in mind as a key player in food and appetite regulation, we found a very interesting study, or in a very interesting study that we conducted, we found that finding here. Oops, oh, now I see it's coming up like this. Okay. We found this finding here, excuse me, where, if you look at these results, these are 4 different samples, all of patients who have been treated with antipsychotics. Patients were not even necessarily all schizophrenia patients, but the weight gain as side effect can occur even in other diagnoses. It's not schizophrenia related, it seems. So in the first result where it says New York, there was a sample from New York, which was actually found by a genome-wide association, pointed out to this gene variant close to the MC4R gene, where people who have this, who are homozygote for this particular genotype, they gained on average 9 kilograms within the first 2 months of treatment, whereas if they didn't have it, they gained much less, on average 3 to 4 kilograms. We then went on and replicated this finding in a sample that we're using here in Toronto, and saw exactly the same effect. Then we replicated this even in a sample that I collected while I was in Berlin. And then we have another sample from Europe, the UFES study, early episode study, where this effect was seen again. So very interesting and very nice replication for one finding throughout very different samples across the world, each of them showing a huge impact of these particular genotype. So that is another gene which is very exciting to look in and perhaps to include in our genetic testing. And by the way, my current NARCET Independent Investigator Award from this year is supposed to look into more depth in this gene. Finally, because we were asked also to give us a little bit of an outlook, what we're doing next, the next larger project we're doing is looking into mitochondrial genes. As you will be familiar, mitochondria play a key role in energy metabolism of the cell and of the organism. And so mitochondrial genes have often not been studied very well, are often not part of larger genetic studies. So we want to go into depth into that. And here's a preliminary finding for one gene in the mitochondria that shows a very nice and significant result where, again, carriers of a certain genotype in this particular marker show on average 4 or 5 times more weight gain than other individuals. And so I would like to conclude my talk in thanking everyone of my group, and please feel free to ask any question, and thank you again for inviting me.
Thank you. Thank you very much for the work you're doing and for your presentation. Our next presentation will be from Dr. David J. Foster, who is a Freedman Prize Honorable Mention recipient. Dr. Foster is an assistant professor of neuroscience at Johns Hopkins University. He's been exploring the neural activity underlying the crucial role of the hippocampus in memory and spatial learning. Dr. Foster.
Thanks. Hope you can see this. So my lab is interested in memory, and that leads us to a structure called the hippocampus, shown here in the human brain and in the rat brain, which is the animal model that we use. Damage to the hippocampus causes severe anterograde amnesia, impaired spatial memory, and the inability to imagine novel experiences. And there are many diseases that can cause damage to the hippocampus, but I draw your attention to schizophrenia, which is associated with atrophy of the hippocampus. Our technique is to actually record from the hippocampus while it's actually performing whatever function it is, by recording from the hippocampi of freely moving animals. And we drop down these electrodes into the cell layer of the hippocampus. These electrodes are special electrodes called tetrodes that triangulate the signal, so it can actually identify different sources of the signal next to the electrode, and thereby isolate the activity of multiple cells close to each tetrode. If you record from a single hippocampal pyramidal cell while a rat is running around in a small arena, 2-meter squared arena like this, you'd see his position over the course of about 20 minutes, given by the black trace, and the single cell would fire an action potential in a very particular way, only when the animal enters a particular region of the environment. What my NASA Young Investigator grant allowed me to do, in part, was to miniaturize the technology so that we could make the tetrodes smaller and the device that holds the tetrodes smaller, so we could actually record with more tetrodes than had ever been used before, and that allowed us to record from more cells simultaneously than had ever been done before. And this is an example of the responses of 212 hippocampal neurons. You can see that there are a small number of cells that we can independently identify as inhibitory interneurons that fire everywhere, but all the other neurons tend to fire in spatially restricted regions of the environment. Each square is a depiction of the entire environment and where the cell fired. And so you can see that across the population, we have something like a map of space. We wanted to probe memory, so we have a memory task where the rats have to find one of 36 wells that can contain chocolate, and he has to get back to it. He doesn't know where the chocolate's going to appear. The task is that he can, there's a single well that we call the home well that's consistently rewarded, and he can find the food there, and then the very next thing he has to do is randomly forage and look for the chocolate somewhere else. He doesn't know where it'll be. He finds it somewhere, and then he can go back to the food well, and he has to randomly forage again, and he finds the chocolate, he can go back. So he's toggling between these two kinds of behaviors. We know he can't smell the food because he can't go straight to the random well. He has to really look for it. And we know he can remember the food because he goes straight back to it. We move the home well location every day, so he has to learn a new memory every day. Now I'm going to show you a few movies. The first movie shows the rat in real time running around looking for the chocolate. What you can see flickering is a representation of the position representation across all of these 212 neurons. So where there's a very bright spot, that's where the hippocampus is saying the animal is located. And he doesn't know where he's going, but you can see that the representation position is very faithfully follows where he is. But now we'll see what happens next. This next movie happened immediately afterwards, But this movie I have slowed down by a factor of about 20. So what you're seeing happen in this movie is 20 times as fast as what happened in the previous movie. Now we see that the representation of position moves smoothly away from the current position and toward the remembered goal position. And this happens many times. Finally, you see that this what we call trajectory event can also depict other trajectories. You see that there are events that move ahead of the animal in a different direction, and in fact, this movie reverts to in real time to show you that this does predict what the animal does. So I'll show you one more movie, which shows again a combination of movement and these internal trajectory events. And just remember that the trajectory events are happening a lot faster than you see, but the movement is real time. So here the rat is looking for the food. He'll find it at the green square. When he does so, he will stop, and the hippocampus will start to generate these internal representations of, in this case, the remembered food location and also another location. Then the animal moves toward the food, and then interestingly, we also see some further predictive activity that seems to predict what his subsequent behavior is also. So you now see he moves off to the lower corner. So this is what we consistently find. To capture this, if you look at all of the trajectory events, this is the internally generated sequences that occur when the animal is positioned at the home well. They have one location in common, which is the beginning location, which isn't that surprising because that's where because there's a bias for trajectory events to start in the current location. But if we consider all of the moments when the animal finds himself in a different part of the environment and look at those trajectory events, they also have the home well in common, suggesting that all these events are approaching this remembered goal. And in fact, we can, merely by examining the trajectory events that are occurring, we can tell where the food is, and this moves appropriately on the next day when we move the goal location. Just one further analysis, we wanted to know if we are really predicting the future trajectory of the animal, and so we've just compared these trajectory events to the actual future path. We find that indeed there's a very strong prediction of what the animal is about to do. So this is the first evidence that hippocampal neurons can encode memory for specific goal locations, and the first evidence that they can predict extensive sequences of future behavior. The task we incorporated several situations where the animal had to construct new trajectories that he hadn't actually done before, and so we also think of this as a new model of memory-based imagination. And finally, I would say, as regards future work, there's a very interesting correspondence between this rest-related activity and recent studies of rest activity in schizophrenic patients, where a network of areas which includes the hippocampus and is sometimes called the default network is particularly active during rest. This is based on fMRI studies, and it's abnormal in schizophrenia. So we're very interested in looking at animal mouse models of schizophrenia to examine this kind of activity. And I'll just thank, in particular, Dr. Brad Pfeiffer, who is the postdoc who did the work, and a NARSAD Young Investigator Award from the Brain and Behavior Research Foundation. Thank you.
Thank you, Dr. Forster, for your work and for the excellent presentation. Our final presentation is from Dr. Hiroki Taniguchi, who is a Freedman Prize Honorable Mention recipient. He is a research group leader at the Max Planck Florida Institute for Neuroscience. He studies nerve cells that inhibit activity in the brain. Dr. Taniguchi.
Okay. So I'd like to thank Brain and Behavior Research Foundation for giving me an opportunity to present my work. We are interested in, All right. So let's start. I'd like to thank Brain and Behavior Research Foundation for giving me an opportunity to present my work. We are interested in how the normal brain works and what deficits in brain functions result in psychiatric disorders. To perform complex neuronal computation, a population of neurons has to work together to generate coordinated the activity patterns. We can measure a population of activity by recording EEG, just like this. Actually, the brain shows different types of rhythmic activity, which corresponds to different psychological and physiological state. For example, when we perform busy tasks using multiple sensory information, the brain shows high-frequency oscillation called gamma oscillations. These activity patterns suggest that multiple neurons cooperate to make rhythms. This looks like neurons play in a symphony orchestra. To be in harmony, you always need a good conductor. So this is also true of neural circuits. Okay. So the chandelier cell qualifies as a conductor because they powerfully inhibit spike generation in the multiple excitatory neurons by innervating axon initial segment, which are sites of spike initiation. So they may control synchronization and oscillation of pyramidal cell activity. So interestingly, there is a link between schizophrenia and chandelier cells. In schizophrenia patients, synaptic markers at axon initial segments are reduced, and GABA neurotransmitter synthesizing enzyme is also reduced. So the current hypothesis is defects in chandelier cells lead to defects in neuronal synchronization. This leads to defects in gamma oscillation, and then defects in the working memory. So, which is a common symptom of schizophrenia patients. So, to comprehensively understand this important class of inhibitory neurons, it is critical to track chandelier cells from birth to maturation. Okay. So during my postdoctoral training at Cold Spring Harbor Laboratory, I identified embryonic the ventral germinal zone, BZZ, as a novel source of GABAergic inhibitory neurons, most of which are chandelier cells, and enabled to observe the whole life of chandelier cells by genetically labeling chandelier cell progenitors. Okay, so in adult brains, we can now reproducibly identify many chandelier cells, which have never been possible until recently, like this. You can see there's a lot of chandelier cells in many areas. Okay, so a year ago, I started my own laboratory and continue to study chandelier cells. So we are pursuing a couple of projects. One is to identify genetic programs that determine chandelier cell fate. So this information may be useful for cell transplantation-based therapy, where many chandelier cells need to be differentiated from iPS cells or embryonic stem cells. So another is to the study development and the function of chandelier cells in neurological disease model mice. These studies will elucidate pathogenesis of several psychiatric diseases. Another is to test if chandelier cell transplantation can rescue the phenotype in schizophrenia model mice. Through these experiments, we hope to find basic biological principles and apply the knowledge to cure for psychiatric disease. Finally, I'd like to thank NERSAT and my former supervisor, Dr. Josh Wang at Cold Spring Harbor Laboratory. Thank you for your attention. That's all.
Thank you. Thank you very much. I appreciate your presentation and each of the presentations. I just have to say the future of psychiatric research with these examples shows how bright the future is. I'd like to now move forward to questions. We've received a few. We will answer as many as we can, and any that don't get answered, Sally Corbett will assist in trying to get the answer for people. The first question that we have is for Dr. McPartlin, and the question is, where do you see going from here with the information that you presented?
Sure. Well, we have a couple of aspirations. The first thing we want to do is, and we actually hope that there is funding from the National Institute of Health to carry out this study. We'll find out in within a few weeks, is to take a look at what we've seen so far in a larger sample of children and to do some different kinds of experiments to understand what piece is eye contact, what piece is eye gaze, and how things like joint attention and displays of emotional expression would be involved. But then we see a few very, very direct clinical applications. So we have ongoing studies of infants that are at risk for autism, and there are a number of experiments that we're currently doing to try to look for biomarkers of atypical brain development before symptoms become evident. And this would be for sure a candidate that we would like to try. We are also interested in using it as a measure of response to treatment. For example, we have pivotal response therapies going on at the Child Study Center that are really designed to improve eye contact and the ability to infer intention from gaze. And we'd like to see if this lets us see who is going to respond to those therapies, and if it also shows us how people are responding to those therapies. And then the other application that we're working on right now is to try to use it as a therapeutic device unto itself. So if we can measure where a person looks in the kinds of these experiments and use where they look to control the experiment, we can also build experiments that shape how a person looks. And so we're building games that respond to a person's eye contact. So you would get points for maintaining eye contact. You would get points for following another person's gaze. And we hope that this could help let us intervene with the gaze patterns of children with autism while simultaneously measuring their brain's learning in the context of these kinds of activities. So those are three of our short-term goals.
Great. Thank you. Thank you. There's a question for Dr. Danese. Do you have any data about whether early treatment, for example, behavioral therapy, following the childhood maltreatment influences the later treatment-resistant depression and/or inflammation outcome?
Thank you. There is no clear data at the moment that early intervention could predict this enduring vulnerability for recurrent and treatment-resistant depression. But this is one of the direction that my research is currently taking. So I will explore whether maltreatment can inform, first of all, stratified medicine, so whether maltreatment can inform whether any specific treatment is more advantageous for some psychiatric patients. But also, as I am a practicing child psychiatrist, I plan to work with children with these difficult histories and present to our specialist clinic in child trauma and anxiety. And I will want to see how treatment of different types, including both psychotherapy and pharmacotherapy, can tap into these very biological vulnerabilities that we have seen are associated with maltreatment.
Okay, thank you. There's a follow-up question related to Dr. McPartlin's presentation, and that is, in addition to the earlier question, I'd like to ask Dr. Borenstein about Dr. McPartland's study. You mentioned that you thought this was very exciting research. Can you elaborate on that? And how could this potentially help clinicians? And the reason that I said it was very exciting research is that we know that early intervention for autism spectrum disorders can make a very big difference for the child. So this research can clearly lead to early intervention and help us have a tremendous impact clinically on how the child does. And obviously, as the research unfolds and they continue their work, there's an opportunity for that to become a part of regular clinical practice. And I'll ask Dr. McPartland if you want to add to what I just said above and beyond my comments.
Well, I would agree with what you just said, and I would add one of the reasons that we're particularly excited about this line of research is we feel it's a fundamentally different way of studying a social disability. Autism it tends to be a problem that exists in the space between two people, and that's a very, very poorly studied space. And so we're hoping that these methods of creating these simulated interactions in the lab are going to, this being the first of many insights that we'll have by looking at autism in the kind of context in which people with autism actually have the most difficulties.
Great, great. I want to ask Dr. Mueller about his work in terms of weight gain and appetite regulation, and what does he see in terms of next steps clinically, given the importance of this issue for treatment?
Yes, thank you. First of all, it is a bit sobering to learn that there is no, no real good antipsychotic in the pipeline in the near future, as far as we can tell, who would just have no such side effect, and therefore, because we will probably have to deal with these drugs for a longer time, we would be happy to have a good prediction of this side effect. And as I explained, genetics can or is looking very promising, and so the pharmacogenetics in general is looking very promising. First genetic tests have become available, even in psychiatry now. Still tests, yes, which need to be further refined, but still already are helpful for patients who have particularly difficult history in drug treatment. And the next step for this particular side effect would be to do exactly the same, to assemble a panel of genes and to basically calculate an algorithm which would then be used as a predictor for patients. And I think that it's fair to say that these tests are currently being developed, and we are collaborating also with others in order to get this achieved. And we hope that we will have soon, first markers being used in the clinical practice, as we did here in Toronto, by the way, for liver enzyme metabolism genes since a few years now. Thank you.
Thank you. Thank you. With time running out, I'll again reiterate that people can contact Sally Corbett with questions. She will be of assistance to, as best as possible, get the answers. And I want to thank and congratulate today's presenters. The Brain and Behavior Research Foundation is committed to alleviating suffering caused by mental illness by awarding grants that will lead to breakthroughs. And I also want to thank the members of the media who have joined us. The media has such an important role in interpreting and sharing the latest information about research, treatment, and care for people, and play an important role in public understanding of mental health issues and research. So I really appreciate folks from the media who have joined us today. The briefing has been recorded, and slides and interviews are available upon request. I also want to invite people to stay in touch with us and explore our upcoming programs. Our next monthly webinar, we have a webinar every month, is on August 13th, and that's with Dr. Carlos Arati from the NIMH, presenting about ketamine and next generation therapies. And we also have on October 25th, our 25th annual New York Mental Health Research Symposium with keynote speaker Dr. Ellen Sachs, the best-selling author and professor of law, psychology, and psychiatry at USC. At the symposium, we'll have a number of our scientists presenting as well. And then the evening of the 25th, we have our annual National Awards dinner where the 2013 Outstanding Achievement Prizes and Productive Lives Awards will be presented. And again, you can see further information about this on our website. Once again, thank you all for joining us, and I'm looking forward to future interactions as well. Take care. Bye.
Thank you very much.
Thank you. And we'll see each other on Friday.
Exactly. Yes. Looking forward to it.
Wonderful.
Take care. Bye-bye.